All right, here we go. Hopefully I can get through the rest of this in one section. Continuing with the middle layer, remember the anterior cavity is in front of your lens, and that area is filled with aqueous humor. Aqua, water, it's a very thin fluid. If you've ever poked an animal's eyeball, if you've dissected the beef eye, that liquid that squirts out is typically aqueous humor. It's very runny. And it circulates from one chamber to the other. It actually transfers through your pupil. And it's also going to leave the anterior cavity and go through the sclera and actually into your venous system. So it's a very thin liquid that transfers very easily. If you've heard of glaucoma, glaucoma is a buildup of aqueous humor, and it actually causes an increased pressure in your eye. If you go to the eye doctor, typically there's a little machine that will like puff air into your eye, and that tests your eye pressure, and too much pressure can cause glaucoma, which if it is severe can actually lead to blindness. So moving on to the inner layer of the eye. The inner layer would consist of the retina, which contains your photoreceptors. It's going to contain your rods and cones that pick up color, pick up shape, and it covers the back side of the eye all the way to the ciliary body that surrounds your lens. In the center of the retina is the macula lutea, which is in the fovea centralis. The fovea centralis is the point in your eye where when it receives images, it's going to have the sharpest vision that there is. So fovea centralis has the sharpest vision. This diagram doesn't concern me too much for you guys. Inside or medial to the fovea centralis is the optic disc. So we talked about the optic nerve being on the outside of the eye. Where it comes in on the retina, there's a small spot called the optic disc, and that's where the nerve fibers are going to leave to the optic nerve. This is your blind spot. So there is a spot in your eye that when it receives light rays, it's actually, you're going to have a blind spot where you don't see a small object. Posterior cavity of the eye, so behind the lens, is filled with vitreous humor. It is much thicker, it's not as runny, it's not as thin, and it has collagenous fibers that form the vitreous body that's in the posterior cavity. The vitreous humor is very important. It's thick, it's going to support, it's going to fill up the eye, it's going to push against the retina so that the retina does not collapse. The retina is a very thin layer, it's easy to tear, etc. So if I'm looking at my eye, the inside, here's that fovea centralis. So when his image is focused there, that's where you're going to have your sharpest vision. And this over here would be your blind spot or optic disc. And here is where your optic nerve would also lead out of that spot. So in order for you to see things, lights have to be refracted, which typically just means they're bent. So they're going to travel through air, and once they hit your cornea and lens, they are going to be bent and refocused on the retina. Also, the vitreous humor and the aqueous humor are going to cause some refraction of those light waves also. If your eye shape is normal, light waves should focus on the retina. It'll be an inverted image. Your brain turns it around. So here you can kind of see, this is a piece of glass. So when light waves travel through air, because the density of air is different than the density of glass, when those waves hit glass, they get bent. When they hit a liquid, they get bent or refracted. And then these waves are going to converge. You want that convergent to happen at your fovea centralis so that you have sharp vision. If you don't have 20/20 vision, if you have glasses, those light waves are not converging at the right spot. So then you have glasses that are bent, so mine are more of a rounded shape. It's going to bend the light so that it does focus in my eye where it needs to be. Two kinds of neurons that are in your retina, they are rods and cones. Rods are sensitive to light. They're going to pick up light and darkness. They only produce black and white images, where rods are not nearly as sensitive, but they are going to produce, oops, I'm sorry, still talking about rods. Rods give less precise images because their axons are going to converge into fewer nerve fibers. They don't have as many. So rods have a less precise image, black and white only. And here you can kind of see a diagram of how rods are more elongated. There's a better picture later. Here we go. So here's an actual magnification. Notice how a cone is more pointed at the top, where a rod is more blunt at the top. They are intermixed. There's not one spot of rod and one spot of cones. So cones, on the other hand, this is what I was trying to skip to earlier, provide sharper images, especially in bright light, and cones color. Cones help us see in color. If you want to see something in detail, you have to move your eye so that that image falls on the fovea centralis. The fovea centralis contains a very high concentration of cones. And then the further out you go from that fovea centralis, there are fewer and fewer cones, and therefore the vision is less detailed or less sharp. Rods and cones both are going to face the choroid and the retinal pigment epithelium that absorb excess light. And we talked about that before. The choroid is black. It absorbs light. We do not want light to be reflecting inside of our eye because that would totally disrupt our vision. Light-sensitive pigment in the rods is rhodopsin. It's a visual, it looks purple. It breaks down into a protein which is just opsin and retinol from vitamin A in light. So light's going to break that down. Decomposition of rhodopsin activates an enzyme that changes in the rod, and that causes the impulse to be sent. So rhodopsin is an important protein in your rods. Those nerve impulses then will travel away from the retina. They go right to the optic nerve and then into the visual cortex in your brain, and that's where you actually see. So your eyes don't see, they pick up light. Your brain is actually where the vision occurs. In bright light, rhodopsin is broken down extremely quickly, and in dim light, it can be regenerated very rapidly. That is another reason we do not want excess light bouncing around inside of our eyeballs. Cones, inside of the cones, there's also proteins and retinol. There's 3 sets of cones, each containing a different type of opsin protein. So what happens is the wavelength of the light that comes in determines the color perceived. Physical science, you've probably seen this. Chemistry, you've probably seen it. Roy G. Biv, the colors of the spectrum. Red has a very long wavelength. Blue has a much shorter wavelength. So the wavelength of that light is how those different pigments are going to work. So then the color that is perceived depends on the sets of cones. If all 3 are stimulated, white is detected. If none are stimulated, black is detected. And then different forms of color blindness result from a lack of different cone pigments. So So if you know someone that is colorblind, their cones are lacking specific pigments, and those can be different. That is why there's different forms of color blindness. Extreme color blindness, you might only see black, grays, and whites. A lot of people have problems with like blues and purples, so there's varying degrees of color blindness. So what happens when you pick up these light waves? It goes to your optic nerve, and then the fibers from each half of the retina cross over. The optic chiasma then forms the optic tracts that go to the thalamus, and there's a picture of this that shows it very well. From the thalamus, nerve pathways called optic radiations end in the visual cortex, which is in the occipital lobe, so the back part of your brain. So here's the diagram I was talking about. Here I have each eye, an optic nerve comes out of the back of each. Optic chiasma is where they actually cross. And then, so this is going to come over to this side, this is going to come over to this side. Visual cortex, where you actually see, takes place at the back part of your brain. So this is the portion of your brain that actually interprets what you see. And that is it for the eyes and special senses.